Lamoureux. I'm a third year PhD student in the Systems Biology Research Group at UCSD. I'm mainly interested in studying genomes and genomic sequences and trying to understand all of the information that they contain, trying to understand how that oldest code that we know of actually works and how it directs the function of organisms from the simplest to the most complex. The genome sequence of any organism contains all the information needed to specify its form and function. And that's true for all organisms. That's true for single-celled bacterium. That's true for a human. But even though we know this, it's actually still really difficult to understand exactly how the genome sequence represents that information. Any particular DNA base, one of those A, T, C, and G letters, can ultimately encode multiple different functions, different pieces of information. So the genome sequence is really convoluted in how its information is represented. It's really overlapping, but yet it is critical to our understanding of an organism's function and behavior. So the goal of this study was to start to try to disentangle that informational encoding, to try to understand how a genome actually stores its information, given what we know about it. So we tackle this challenge by essentially digitizing the genome. What does that mean? Well, for any given DNA base, one of those letters, A, T, C, and G, we can ask a number of yes and no questions. We can ask, is this base involved in coding a gene? Is it involved in coding for a specific amino acid within that gene's product, et cetera, et cetera? Is this gene regulated in a certain way? And the answer to all those questions is a yes-no answer, and that's a binary digit, a 1 or a 0. So we can move through the entire genome sequence, one base letter at a time, and ask those questions and record a 1 or a 0 depending on the answer. And in this way, we actually have a binary representation of information that is encoded in the sequence. And this collection of bits for binary digit, we call the bitome as a homage to the genome on which it is based. And this new bitome representation of a genome is great because we can do computations with it. It's a quantitative object. So instead of having information kind of stored in text between different databases, we actually have just one binary representation in a quantitative way that we can use. And so in our case, we've used that to demonstrate a couple of interesting results. First of all, we showed that genes with fewer bits of information, so genes or even just specific DNA bases with fewer yeses as answers to those questions, are more likely to be mutated during adaptive evolution, which suggests that they are less important, perhaps, based on encoding actually fewer pieces of information. Additionally, we were able to use this bitome representation for machine learning. Machine learning methods love quantifiable inputs. They don't work so well unless you can convert your inputs into a kind of numerical format. So this way of encoding genomic information works really well for machine learning. And we were able to show, we were able to predict the mutability of genes as well as the essentiality of genes based on the accumulated information across their genome positions, the bases in those genes. And additionally, those machine learning models revealed that certain unexpected relationships support that prediction. So for the mutable genes, a certain stop codon, UAG, is actually associated with increased mutability, while in the case of gene essentiality, we showed that a predicted residue exposure of the protein encoded by a gene is also predictive of essentiality. So the bitome is useful not only as a way to study the information contained within a genome, but to make predictions based on that information. So this study provides really an entirely new way of analyzing any genome sequence. We can extend this method to any organism's genome. We just have to map information that we know about the genome sequence onto each base of the genome, ask those yes and no questions. And we can do this even without experimental information. There are many properties of a genome sequence that can be calculated just from the sequence itself. And those are also useful for understanding how the overall genome's information is encoded. So in this way, the Bitome helps us with a deeper understanding of information encoding in a specific genome, as well as enabling prediction based on that information encoding. So therefore, with this work, we really hope to further bridge the gap between just having a genome sequence and really understanding how the information in that sequence is acting and is functioning.